Hello, and welcome to this cell division overview. At the end of this video, you will be able to describe the steps of cell division. Cell division naturally occurs as a continuum, which we mentally divide into steps so we can keep track of what's going on. Importantly, most cells are not actively dividing. Division is one function that cells can go through, and some cells divide rapidly and frequently, stomach cells, for example. Other cells hardly divide at all once the organism has reached maturity, neurons, for example. So if we look at the number of cells that have replicated their DNA and therefore are in the process of dividing, we see that the majority of cells have one copy of DNA, which means they are not dividing. And a smaller fraction of cells have replicated their DNA, which means they are actively undergoing division and have not yet divided. The cell cycle itself is divided into 4 stages, which are regulated by checkpoints. So the first of those stages is growth 1. G stands for growth. And before the cell will enter into S phase, it will ask itself through a series of molecular checkpoints whether or not the environment is favorable for division and the cell is big enough to hold a whole extra copy of DNA. If the cell passes those checkpoints, it moves into the synthesis phase. This is when DNA replication occurs, or when DNA is synthesized. Once the DNA is synthesized, the cell undergoes a second growth phase. And before the cell can move into the final phase, it asks itself whether all of the DNA is replicated, whether any DNA is damaged, and again, whether the cell is large enough to support the division into 2 smaller cells. That division occurs in M phase, which generally stands for mitosis, although as you may know, some cells are actively undergoing meiosis to generate gametes. There's a separate checkpoint during mitosis, mostly during anaphase, where the cell ensures that all chromosomes have been attached to mitotic spindles and are therefore going to get pulled apart into their respective daughter cells. We'll look at that in just a second. So the M phase is divided into even further steps, which are shown here and numbered: prophase, prometaphase, metaphase, anaphase, and telophase. And we will look at each of those in turn. Interphase is the start of cell division, where the cell increases in size. This includes parts of S phase, where the DNA itself is replicated, and the centrosomes are duplicated because they will be necessary for microtubule formation during anaphase. Once the DNA has been divided and the centrosomes have been replicated, the cell moves into prophase. During prophase, the nucleus itself dissolves and the DNA becomes condensed from its kind of heterochromatic and euchromatic forms to the mitotic chromosomes that we're used to seeing. Additionally, new mitotic spindles, which are made of microtubules, start to form from the centrosomes, which move to the poles of the cell, preparing for division. In prometaphase, the nuclear envelope finishes its dissolving, and the chromosomes begin to attach to mitotic spindles using their kinetochores. So each chromosome has a kinetochore, and the microtubules are being grown from the centrosomes. So these are microtubule organizing centers, organizing the minus ends of those microtubules, which are growing and shrinking, attempting to capture chromosomes here at their centromeres. As it's doing that, it's essentially pushing and pulling the chromosomes so that they can align in metaphase. In metaphase, the chromosomes themselves are aligned at the center of the cell. Remember, it's not the center of the nucleus because the nucleus has dissolved. So at approximately the midway point of the cell, the chromosomes themselves align, and they are still attached to microtubules, getting ready for division. At this point, there's a checkpoint where the cell ensures that each chromosome has in fact been bound by a microtubule and that the chromosomes themselves are aligned such that if we divided the cell down the middle, each daughter cell would get an equal number of chromosomes. That division happens in anaphase, where the sister chromatids, that is the replicated copies of each chromosome, are pulled apart toward their individual spindle poles. So the microtubules are getting shorter, and as they do, they pull the chromosomes toward the poles, toward the centrosomes. Once the chromosomes have been separated, the nucleus reforms around each of those sets of chromosomes. So 2 new nuclei are created. So the division of chromosomes into new nuclei occurs in telophase. And the final division of the cell occurs during cytokinesis. Cytokinesis occurs when actin cytoskeleton is arranged around the contractile ring, that is around the center where the cell is going to divide. And then as that actin contracts and gets shorter and shorter and shorter, eventually there are 2 fully distinct cells created. So the act of dividing the cell into 2 separate cells is cytokinesis. What we'll see in a future video is that these molecular mechanisms of cell division rely on many proteins and processes that we've discussed in the past.